Hi, welcome back. This should be the last video for the series to look at the pathway analysis and how we can actually use the data from DRIVE to identify the candidate drugs for further testing. Okay, so in the past several video, basically we have explored how we can use the Drive dataset, download the Drive dataset, and then extract the information. We have also covered, for example, how to do the normalization using the shRNA-seq. Then, okay, in the most recent one, okay, I also show you how to run to filter the data using the Z-score cutoff, RSA, also the rigor analysis. Now that we have all these okay filtered genes, so which each of them could be a candidate hit, but in order for us to actually identify the drug targets, it would be good that we can actually collapse all these hits into a possible pathway and also suggest certain drugs that we can test. In order to do that, basically in this video, I'm going to cover okay the three things here, including to calculate the mean Z-score. Okay, then after that we will use the top 100 genes from the mean Z-score and then to construct the pathway analysis and also carry on to identify the drug targets. Okay, so I'm just going to use the file that you have uploaded to Dropbox. Basically, what you have here is you have the original Z-score. This is after the shRNA-seq normalization, and you also collapsed them into the calculated the mean for each of the genes. Z-score, which will be very useful, and you also conducted the Z-score cutoff, the rigor, and also the RSA analysis, and these are the genes or the candidate hit that are basically identified for each cell lines. So what I'm going to do now is basically I'm going to create the list for all the genes that you have filtered. Basically, this will be the hits for all the cell lines. So I'm going to create that list here. And all you need to do is basically you are going to copy each of this list here into a separate sheet and then remove the duplicate. Okay, once you have removed all the duplicate and also the redundant genes, you should have about 2,847 genes, which is identified as hit from each cell lines. Now, what I'm going to do next is basically I'm going to compile this table with all the cell lines and also the average Z-score here by using the VLOOKUP formula again. So, goes to VLOOKUP. So this will be my lookup value, and then the range that I'm going to look up for would be from here all the way to the end. Okay, and that will be returned from column number 3 and 4. So then you should get okay the value here. So this is basically the this is basically the mean score for this particular gene of in the BSPC3. So what you're going to do next is basically you can then use the same formula then to complete the whole table here. So after it is completed, you should get a table something like this. So what I'm going to do now is basically I'm going to calculate the mean across all the genes here. So you can see that for the highest, okay, for SNRPD1, for example, okay, the mean Z-score across all the cell lines here are basically minus 4.7. So what I'm going to do next is basically I'm just going to sort it from the smallest to the highest, meaning that basically the lists here are basically sorted by the most potent gene knockdown in all the cell lines in the list that we actually identify. And here what I'm going to do now is basically I'm going to just select the top 100 genes here, okay, and then I'm going to put this list into the pathway analysis. So for pathway analysis, what I'm going to do now is basically we will use the online software called String. So you can read up about the basic concept about string. So basically what you need to do is that you put the input list here. Again, this is a top 100 genes that when knocked down induce a significant amount of uh, self-growth inhibition in the pancreatic cancer. So we are going to select the organism is Homo sapiens. Okay, so then you press continue here. And what it does is basically it will actually generate a network map to show the relationship between each of the candidate genes that you actually identify. So, and each of this color line here basically represents something. You can look at the legend here. It will tell you what is the relationship that is related from one gene to another. So you can, as I said before, you can read up more about the string algorithm. It's actually quite interesting. If you want, you can also move the node as you like. Basically, it's an interactive map here. Now, what I'm going to do next is basically I will go into the settings. So instead of showing all the evidence lines here, I'm just going to be interested to show the confidence line. Okay, basically in the confidence line, if I update it, what you will see is basically the darker the line is, the higher the confidence it is. Now, so you note that basically the required interaction score by default is set to 0.4. So in this case here, I'm going to increase it to the highest. Okay, only 0.9, and I'm going to update the map. So now you can see that, okay, less of the node are actually connect to each other. You have a lot of the node that do not connect. So now, so what does all this mean is basically you can also go into the analysis here. For example, it will straight away tell you, okay, what in the CAC, based on the CAC pathway, which of the pathway are particularly enriched by the gene set that you put it in here. In this case here, okay, you have 10 genes, okay, in your list, which is within the spliceosome pathway. And when you select this one here, it will highlight what are those genes. You can see that this is the group of the genes that are related, okay, in the spliceosome pathway. And then if you look at the second highest and significance is basically the proteasome, the enrichment. Significantly is a proteasome pathway here. And then same go for synaptic vesicles, okay, and also the ribosome pathway here. So the rest of it doesn't seem to be enriched particularly significant in the CAG pathway. So in this case here, we will conclude that basically the top 100 candidate genes that you identify from the THRIVE study basically are enriched in the four pathway here. What it means here is basically, okay, by inhibiting the spliceosome, proteasome, or ribosome, and also the synaptic vesicle cycle, there might be a high potential that you would be able to target the pancreatic cancer. Okay, so now that we have identified the four pathways that might be potential for targeting, the next question is basically what are the drugs that we can use to actually target this pathway and also the top 100 genes that we have identified. So what we are going to do now is basically we are going to use another online database called the DGIDB. So this is basically the drug gene interaction database. So what you need to do is basically you can search the drug gene interaction by clicking this button here. All right. Then you just need to put in the gene list. This is the top 100 gene list that you have identified. And then you click on the find drug gene interactions here. Okay, so what you can see is that for the gene SNRPD1, there's no known drugs that is targeting it. So same go for PRPF8, for example. These are the genes that have no target to it. So RAN, you have one drugs that could target it, and if you click inside here, they will tell you the information about this particular drugs, basically. Now, but what is interesting here is basically you can see that PSMA4, which is a proteasome inhibitors, you have a range of proteasome inhibitors that could actually target the PSMA4